There we go. Hello, biology. Welcome back. Welcome back to part 2 of talking about our notes for cell replication. Last time we talked about binary fission and mitosis. We also talked a little bit about spontaneous generation as well. If you remember from last time, we last talked about interphase and what the basics of it was, including things like, the checkpoints and the little parts of each, the parts of interphase that can happen. Okay, so here's a review. In interphase, our DNA, in this case, on our picture, is blue. It's in its chromatin phase. As you can see, there's not really any big details. You can see it's just kind of a big blue blob. Okay. We do have a nuclear membrane present. You can't really see it, but it's basically kind of like that white border that surrounds the blue. Okay. And that's because the chromatin is inside of the nucleus. Okay. It asks, do you see centrioles? Centrioles are the little things that help to pull the help with the cell division. Okay, and they are in green on here. Okay, so reviewing what interphase looks like on a slide in real life, what it looks like if we used colors to enhance it. So as a review, interphase is that phase that we have before we do any kind of cell division. It's our growth phase, and so this is where most of our cells kind of sit, is in this mode. Okay. As we go on, mitosis is split up into different phases past interphase. Okay, and the first phase of mitosis is called prophase. Okay, prophase, a couple things happen. Now, before I keep going, I want to make sure you can tell that I have pictures here and a list of things that happen in each. For this unit, I do expect you to know the names of the phases, number 1. Number 2, what happens in those phases. And number 3, what that phase looks like. Okay, so I expect all 3 of those things to be known. The name, what happens, and what it looks like. Okay, so continuing on, the first phase of mitosis, we're talking about the specific phase of mitosis now, which is this phase right here in the cell cycle. Okay, we're talking about the actual division process. First phase is prophase. This is where our chromatin coils up into our chromosomes, into our X-shaped things. Remember us talking about that last time? Your nuclear membrane around your nucleus disappears. And then you have the centrioles in the cell move to opposite sides of the cell, forming spindle fibers. That's why the green thing is like spidery. And then the chromosomes begin to move inside of that, what used to be the nuclear membrane, as it disappears. Okay, so it goes from this, which was in an interphase, to this. As you can see, the blue blob in the middle is no longer just a straight blue color. It also has it looks like darker blue dots in it. This is showing that our chromatin is coiling up into chromosomes. And then the centrioles here, you can see, are becoming more defined. There are 2 of them, and they're moving away from each other. Whereas over here in interphase, they were kind of together, overlapping each other. Okay? After prophase comes metaphase. the way I remember metaphase is it starts with an M, and M stands for the middle for me. It's like in the middle of everything from interphase to the finish line. It's right in the middle. Okay. This is where our spindle fibers attach to our centromeres. Remember, our centromeres are the center parts of our X where those two chromatids meet. The spindle fibers, the green spindly, spidery, spider web looking things will attach to those middle parts, okay? And then chromosomes line up at the equator of your cell. So it goes from this to this. You'll notice your blue looks more like noodle threads or your X's rather than as a big blob, okay? And you'll notice all the blue is lined up in the middle. You can see your green threads here, which are your spindle fibers, attaching to those blue chromosome centromeres in the middle. Okay, that's another reason why metaphase stands for middle, is because the chromosomes line up in the middle of the cell. Okay, metaphase, middle. After metaphase, there's anaphase. Your centromeres split, as well as your sister chromatids. They get pulled apart. So your X starts as your X looking chromosome, and each side is a chromatid. What happens in anaphase is that it's kind of split down the middle and then pulled apart. Okay? This is why anaphase starts with an A. It stands for the word apart. Okay? Because that's what happens. We go from this, where metaphase had everything lined up in the middle, and things were still together, and anaphase is where, see, our green centromeres and our spindle fibers are pulling up, going this way for this one, and this one is going that way. So this one is going this way, and this one here is going that way. And your chromosomes, which were together in the middle, are now being pulled apart in separate directions. Okay, following the centrioles that they're attached to. Okay. So, anaphase is for apart. After anaphase, we have telophase. This is where our chromosomes uncoil back into chromatin. They relax. The chromosomes go, whew, we're done, and they just kind of relax. Your nuclear membrane starts to form around your new bunch of chromatin. That's the chromosomes that are relaxing. Your spindle fibers start to go away, and your cytoplasm begins to divide. So in anaphase, we had our chromosomes being pulled apart, going to its new location on each side of the cell, and telophase is where we're starting to pull, like fully tear apart. So telophase, the T stands for tear. Tear or tearing. Because that's what's happening in telophase. The cell is beginning to divide and tear apart. Okay? And this is technically the last phase of mitosis. Okay, there are 4 phases of mitosis. Starting from the beginning, it's prophase, metaphase, anaphase, and finally telophase. Okay, those are technically the 4 phases of mitosis. Once you finish with mitosis, we go on to the next phase, and we'll talk about that in a second. I have 2 things for it. One is a video, the other one is just I'm going to look at a couple things. The ultimate result of mitosis is 2 copies of the exact same cell, or daughter cells. Ding dong, hopefully you said that looks familiar. The reason it looks familiar is because this was the result, or the whole goal of the other type of replication we looked at, which was binary fission, creating 2 cells that were copies of each other. Okay, they were clones or the exact same. Okay, all right, let's look at that video, which goes over mitosis as a whole.
Mitosis begins as the long threads of DNA in the nucleus start to coil.
Prophase.
Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates.
Still prophase.
With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell.
Still anaphase.
The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating 2 new cells in this ongoing cycle that sustains all life.
The last bit here before we end, right there, as it was going to, it was, notice it's starting to it's starting to fold, or it almost looks like close off here. This is going to be the last phase
all life,
afterward, which is called cytokinesis. Okay. Here's a website that shows you division. It goes over the overview of it, and it shows you the cell cycle again. So this is a good place to see your pictures in a different way. So prophase, metaphase, in 2 different, so this one's a plant cell, this one's an animal cell. So prophase, metaphase, anaphase, and then telophase. And then after we have mitosis, we have cytokinesis, which is our actual separation of the 2 cells from each other. And then it shows at the end here interphase. So when we start when we go into the growth and whatever phase. Okay, so if you need to see those in a different way other than what's on my slides, they're here for you. Okay. So, yep. So our whole result of mitosis is our 2 exact same cells which is good, right? Because you don't want your skin cells that are dividing to become something else. You don't want them to become heart cells all of a sudden, right? You want to have them be the same type of cell, which is skin cells, on your skin, right? You don't want some muscle cells or something on the where your skin should be. So that's why mitosis is important. Okay? All right. After telophase, we enter our final phase of our cell cycle, which is cytokinesis. And this is where cytoplasm just divides, resulting in our two cells. So in animal cells, our membrane pinches from the outside in, kind of like if you took a string on the outside of like a water balloon and you tightened it, that you kind of get that look. Or like if you did it to jelly, you know what I mean? If you took Jello and you put a string around it and you tightened it until it pinched off from each other, it makes two separate pieces of Jello. That's what cytokinesis is like. It just takes the cell pinches off with like almost like an invisible string like instinct, and it pinches off. Whereas in plant cells, they actually form a plate on the inside and goes outward. And this is just to keep it from spilling out, because animal cells are kind of squishy, as all like us who are kind of squishy like beings. It's okay if we have our cells pinch off to keep all the squishiness inside, whereas plant cells are really rigid and they don't want to have their insides of each cell spill into each other because they're rigid. So instead they make their barrier wall inside before they split off from each other. Okay, so just know that each cell, when they do cytokinesis, animals pinch from the outside in, plants separate from the inside out. And do plants go through mitosis? You betcha they do. Yes. This whole process that we just saw, this whole prophase, metaphase, anaphase, telophase thing, we do it, and so do plants. They do the exact same thing we do. Okay? And we can actually see that in our cells of some plant roots, because that's where mitosis is the most prevalent, is in the roots where there's active growth happening. So when you look in closer, you see onion roots, for example, that look like this, and you can tell which phase those cells were going through at the time that the root was looked at and preserved for looking at. So this one is probably in either interphase or prophase, and you can tell that because the nucleus is still a bunch of, it looks like noodly stuff. It's just one big color. This one, I would say, since everything's lined up in the middle, this is metaphase. You can kind of see the spindle fibers. Let's make them green because that's what they were before. You can kind of see them like here, coming off of your chromosomes, getting ready, like that on that side and like this on this side. You can see it. And so you can assume like the centromere is like in this area here or in like this area here. And then for this one here, it looks almost like it is in either it's in telophase. And the reason you can tell that is because your inside of your nuclear membrane here that is forming, nuclear membrane is right here. And inside it, you don't have it look like this, which is your relaxed chromatin. It's still kind of in like chromosome form, you know, the little bar forms, which indicate that it's either early telophase or late telophase. You can see that a divide here is starting to maybe form. So we're probably in late telophase or very early cytokinesis for this one. Do I expect you to do this on your own and be able to tell different cell-like phases apart? Yes, I do. Absolutely. Here's a couple more examples in order. So prophase, or interphase, sorry, interphase. So this is interphase. Prophase, and the reason you can tell this is prophase here is because you don't have a nuclear envelope anymore and you have your chromosome, well, it's starting to dissolve and you have chromosomes instead of chromatin. This is metaphase because you have stuff lining up in the middle. This is anaphase because things are getting pulled apart. And this is telophase because things are starting to reform. You're looking like you're going to get some, you know, two separate cells again, but it's not quite there yet. It doesn't have that hard line between cells quite yet. Okay, so that's what the cell cycle looks like in onion roots from interphase through telophase. Okay. And do I want to keep going? Yeah, I'll keep going. So when this whole cycle here is messed up and there isn't any way for there to be any regulation, that's when mitosis goes wrong, when either your DNA doesn't replicate correctly, the cell doesn't have the right instructions anymore. It can lead to uncontrolled replication where mitosis happens over and over and over without stopping, without checkpoints, nothing like that. It can lead to cancer where our cells don't follow our cell cycle properly anymore and simply divide without stopping. And this is why tumors, when you have them, can be called benign or malignant. Benign is where there is some sort of semblance of the tumor stopping its growth, whereas malignant is where the cell cycle copying is on loop continuously, no stops, no pauses. Okay? Okay. And that's where I'll stop. That is the end of part 1 of talking about cell replication. We've talked about today mitosis, the phases of it, what happens in each phase, and what each phase looks like. We had our interphase, which comes before mitosis, and then we talked about mitosis, the 4 phases of it, which is prophase, metaphase, anaphase, and telophase. And then we talked about our final phase of cell replication, which is cytokinesis, or where the 2 cells that were being made are basically cut off from each other to make them independent cells. Okay, and then we lightly touched on what happens when mitosis goes wrong and how we get cancer. Okay. Coming up next after this, we switch over to part 2 of our notes and we start talking about meiosis, which is another process which talks about sexual reproduction instead of asexual reproduction. Okay? And we'll talk about that next time. But for now, I'll see you next time. Take some time to look at your notes again if you were lost on anything, and let me know if you have any questions on what we've covered so far. Okay? All right. I'll talk to you all later. Take it easy. Bye.